Hi, we are Team 4. In this video, we'll explain the microscopic characteristics of MRSA, the pathology it causes, and how MALDI TOF mass spectrometry enables rapid detection. Well, methicillin-resistant Staphylococcus aureus, or MRSA, is a major threat in healthcare settings. It frequently causes skin and subcutaneous tissues infections, followed by invasive infections like osteomyelitis, meningitis, pneumonia, lung abscess, and ineffective endocarditis is associated with an increased morbidity and mortality compared to any other organism. Under the microscope, MRSA appears as coxine clusters, sometimes changed. They are gram-positive, meaning they retain a violet stain due to their thick peptidoglycan cell wall. They are 0.5 to 1.5 micrometers in diameter and may produce a golden pigment called staphyloxanthin. MRSA is defined by its resistance to methicillin and other beta-lactam antibiotics due to the presence of MALDI gene, which encodes for PBP2A, an altered protein with low affinity for these antibiotics. This resistance can be transferred between bacteria via bacteriophages. MRSA can asymptomatically colonize the skin and nasal passages of individuals. Plus, hospitalization increases risk, especially after surgery or with the presence of invasive devices or weakened immunity. After exposure, the incubation period typically ranges from 4 to 10 days. During this period, MRSA may colonize the host without causing symptoms. After the incubation period, the prodromal period is presented, where initial signs may include small red bumps resembling pimples, which can be accompanied by fever and general discomfort. The bumps can quickly evolve into painful abscesses filled with pus. Then the infection can penetrate deeper into the body, leading to several conditions mentioned before. Infected individuals may 1. Recover, depending on the severity, 2. Develop chronic infections if risk factors persist, or 3. Die, especially in cases of sepsis. With serious infections, every hour matters. MALDI TOF MS offers fast, accurate microbial identification by analyzing protein profiles. While not designed for one specific pathology, MALDI TOF is used to identify the causative agent and resistant markers. So, how does it work? First, a pure bacterial culture is needed, grown on a standard media like blood agar or tryptophan soy agar. Depending on the study, bacterial samples may come from nasal, throat, or skin swabs and are sometimes frozen before analysis. In most protocols, a small amount of bacterial colony material is carefully transferred onto a designed spot on the MALDI target plate. However, some studies also include extra protein extraction steps like centrifugation of the culture, resuspension of the bacterial pellet, and distilled water or heat inactivation. Next, a special chemical solution called the matrix is added on top of the sample. The matrix is typically a saturated solution of alpha-cyano-4-hydroxycinnamic acid in a mixture of acetonitrile and trifluoroacetic acid. As the liquid evaporates, the sample and matrix co-crystallize and is located into the mass spectrometer. Once inside the mass spectrometer, a laser is directed at the co-crystallized spot. This matrix absorbs laser energy and transfers it to the bacterial molecules, facilitating both their disruption and ionization during the laser pulse. The lasers commonly used for MALDI are pulse ultraviolet lasers. These matrices often have conjugated aromatic systems, which absorbs UV light efficiently. Laser pulses are extremely short, just 1 to 10 nanoseconds to minimize thermal degradation of the analyte. As the matrix absorbs the laser energy, it heats rapidly and undergoes sudden expansion, generating photochemical stress that helps eject the sample into the gas phase. This disruption is coupled with a cascade of high-energy collisions and rapid thermal changes. In this energetic plumb, ionization takes place. The matrix acts primarily as a proton donor, transferring protons to analyte molecules, resulting in positively charged ions. These ions are then accelerated through a vacuum tube by an electric field, which gives them the same kinetic energy. Lighter ions travel faster down the tube, while heavier ions travel slower. That's the time of flight part. The detector measures the arrival time of each ion. The instrument generates a spectrum, which is essentially a graph showing the intensity of detected ions versus their mass to charge ratio. This spectrum represents the unique protein profile of the bacterium. This protein fineprint is compared against a database for identification.
To detect MRSA, we look for specific molecules associated with methicillin resistance, While the crucial PBP2A protein itself or its fragments are not typically detected by this specific method, other important molecules found in many MRSA strains can be. One key target molecule is phenol-soluble modulin MEK, or PSM-MEK. This is a small peptide toxin encoded by a gene often found on the same genetic element that carries the MEK-A gene in certain MRS types. It plays a role in promoting inflammation and cell lysis, including the lysis of neutrophils. It is also involved in biofilm development, which enhances bacterial adherence to surfaces and resistance to antibiotics. PSM-MEK shows up as a characteristic peak at 2415 mass charge radio in the spectrum. PSM-MEK gene is located in the complex MEK-A class A within elements of SCC-MEK cassette, which are mobile genetic elements. So PSM-MEK gene promotes the production of the PSM-MEK protein that is an amphipathic alpha helical peptide that allows MRSA to integrate into and disrupt cell membranes. The instrument software automatically analyzes the spectrum, often specifically looking for these marker peaks, along with the overall protein profile for species identification. Manual analysis can also be performed. On average, results can be generated in just 6 minutes, compared to 5 to 48 hours required by conventional identification methods. To ensure accurate results, different controls are used throughout the process. A bacterial test standard provided by the manufacturer, such as the Bruker-Daltonic standard, is used for calibration, and some methods use an internal peak at 4306 mass to charge radio as a reference. For MRSA detection, MRSA N315 can serve as a positive control since it produces the PSM-MEK peak. As a negative control, MSSA ATCC 29213 can be used, showing no such peak. Results from the MALDI TOF test for MRSA are typically reported as positive. The spectrum shows S. aureus protein profile and includes the specific peaks associated with methicillin resistance like PSM-MEK. Negative, the spectrum may show S. aureus but lacks the characteristic resistance peaks. Doubtful, the spectrum may be unclear, incomplete, or show ambiguous peaks requiring further evaluation or repeat testing. Studies show that MALDI TOF MS is highly effective for identifying MRSA. It shows a sensitivity of about 92% and a specificity of 97% compared to clinical reference methods. MALDI TOF can be used either as a screening tool or a confirmatory test. Given that it provides rapid identification and strong evidence for resistance based on protein markers, it's generally considered a powerful tool within the confirmation process. Sometimes, particularly in ambiguous cases, further testing like PCR targeting the MEK-A gene may still be used for absolute genetic confirmation. MALDI TOF MS offers several key advantages for MRSA detection. It delivers identification and resistance information much faster than traditional culture and susceptibility testing. It's more time efficient than methods like MLST or PFGE for typing. It's considered more cost efficient per test than PCR once the initial instrument investment is made. It's more discriminative for strain typing compared to methods like SPA typing or basic resistotyping. From a pure culture, sample preparation is relatively straightforward, just transferring the colony and adding matrix. It can even be used to subtype specific MRSA lineages like CC398. However, like any technology, it has limitations. Some studies point to lower repeatability and reproducibility compared to other methods. Factors like sample preparation consistency can influence results. One key challenge is the formation of matrix-derived ions and the composition products. These create a non-specific chemical background that can mask or distort the true analyte signals, particularly in the low to intermediate mass range. MALDI TOF systems are increasingly used in clinical microbiology labs. Various laboratories and companies offer this technology and testing services. For example, companies like Biomic and DNA Labs India are among those providing or utilizing MALDI TOF solutions, and InnoLab offers the MALDI TOF BioTopper system. While the initial instrument cost is significant, the cost per test is relatively low. Depending on the setup, reported costs range from approximately $0.5 per test to about $1 per minute of machine time. In conclusion, MALDI TOF mass spectrometry is a powerful tool revolutionizing clinical microbiology, particularly for rapid detection of challenging pathogens like MRSA, by quickly providing a protein fingerprint and identifying resistance markers like PSM-MEK. It enables clinicians to make faster, more informed decisions about patient care and infection control. As technology advances, MALDI TOF is set to play an even larger role in combating antibiotic resistance and improving patient outcomes.